Hello, my name is Kevin Cash, and for the last few years I've been working on completing my thesis project of how Chelonibia testudinaria, a species of turtle barnacle, allocates its resources, as well as how this epizoic barnacle exhibits phenotypic plasticity whilst living on the backs of a mobile host. Before I start, I would like to quickly say that all photos displayed within this presentation were obtained by authorized personnel on FWC MTP 255. Only authorized personnel were allowed to work on or with any turtles, as well as any samples collected thereafter. So this brings me to a brief overview of how I will be defending my thesis today. Starting off, I will be giving some generic background information regarding barnacles in general. From there on forward, I will be getting into more background information regarding my project, as well as specifics about the target species of barnacle being observed within this research, and that species, of course, is Chelonibia testudinaria. Continuing on, I will be providing the questions being asked throughout this research, and the significance behind their potential findings. From there, I will be discussing the application of the research, going over the sampling and laboratory protocols, as well as the statistical tests that were conducted, and these protocols then yield results, which have been separated into how the barnacle's penis and cirri morphology were affected, fertility probability, and spatial distribution. Followed thereafter is the conclusion and significance of my research, finishing with the acknowledgments and references. So, barnacles are ancient organisms that date as far back as the Middle Cambrian period of the Paleozoic era, and there are about 1,400 living species found all over the world, covering from the deep benthos to intertidal zones. And barnacles were originally classified as mollusks due to the appearance of the animal, and this was due to the calcareous shell, referred to as a carapace, resembling the outer shell of a mollusk. There was also an apparent lack of segmentation, and Jean-Baptiste Lamerick suggested in 1809 that barnacles were in fact not mollusks and should have their own class known as Cirripedia. And in 1830, barnacles were then reclassified by John Van Thomson as crustaceans, and the defining characteristic that ultimately differentiated barnacles from mollusks was found within their larval state. Barnacles molt, have jointed limbs, and even have similar jaws to those of crustaceans. Following this discovery, barnacles were officially classified as Cirripedia under the subphylum Crustacea. The calcareous shell completely encompasses the innards of the organism, providing defense and structural stability. And the carapace is comprised of a few important pieces: the rostrum at the anterior portion of the organism, the carina at the posterior portion of the organism, and the rest of the barnacle is surrounded by lateral plates. The operculum, which is the opening, is protected by two different parts, the tergum and the scutum. Within the protective housing, a soft body exists. And some key features include the feeding cirri, to which there are 6 on both the left and the right side of the soft body tissue. And each cirri is split, branching into 2 identical structures. The length of the cirri increases from 1, being the smallest, to 6, being the longest. The cirri are also unique to one another, in the sense that they perform different jobs for the barnacle. Cirri 1 is primarily used for grooming or holding on to food particles, and the other longer cirri are used for filter feeding, or collecting food particles and bringing them inside of the shell for consumption. Barnacles also have a penis, and their penis is located in between cirrus 6 left and cirrus 6 right. So barnacles are simultaneous hermaphrodites who reproduce by means of pseudocopulation with neighboring barnacles, and most barnacles are sessile beings, making it difficult for individuals to fertilize egg masses. Barnacles combat this issue, however, by means of their extremely long, flexible penises. Barnacles have the largest penis proportional to the body size of the entire animal kingdom, and a barnacle's penis has a series of small segmentations known as annulations, and these annulations are folds in the cuticle of the penis that allows for it to extend in search of a mate. Once a functional male has found a functional female, sperm is released from the penis and deposited inside of the mantle of the receptive female. And after the mating season, the penis is then typically shed during the first post-mating molt. And barnacles are unique in the sense that they exhibit several different sexual systems. And those systems include simultaneous hermaphroditism, andriosy, and diosy. Simultaneous hermaphroditism is when an individual has both male and female reproductive systems. Andriocy refers to a population where one can find both hermaphrodites and strict males, and diocy is when a community is comprised of strict males and strict females. Whether strict males make up a community of barnacles, they are always smaller than the rest of the population they reside within, and they are referred to as dwarf males. As I'd mentioned on the previous slide, all strict males are smaller, referred to as dwarf males. However, they are not to be confused with complementary males, as complementary males are small hermaphroditic barnacles that act as males during the mating process. Dwarf males are non-hermaphroditic males. Dwarf males are also commonly found growing upon the outer shell of larger hermaphroditic barnacles, and dwarf males act as a potential sperm donor for the larger individual it grows upon. Dwarf males typically only live for a brief amount of time, and dwarf males tend to do well in smaller mating groups, typically found in deep sea environments or when the barnacles are attached symbiotically. They do better in these environments because the sperm competition is intense between hermaphrodites and the smaller individual males, so the smaller the community, the better the dwarf males can successfully reproduce. Dwarf males do have a few advantages over complementary males, and these advantages include earlier maturization, allowing for reduced generation time, as well as the obvious advantage of growing directly on top of a potential mate. Growing on top of a larger hermaphroditic barnacle increases fertilization success per sperm, and in some cases, if there is not enough successful fertilization between hermaphrodites, the hermaphroditic barnacle's male function can be lost permanently. This then causes the community to rely solely on whatever dwarf males are within close proximity. However, hardly any dwarf males were collected within this research. Understanding potential impacts dwarf males can have on a community is still a vital part to barnacle ecology. So, phenotypic plasticity is the ability for an organism to change in response to inputs from the environment. The same species found in one location are morphologically different from those found in different locations with a different set of variables. And this change may involve its morphology, physiological state, or even behavior. And a few examples of influential factors include host origin, substrate type, seasonality, and laboratory or field conditions. When barnacles are exposed to heavy wave action, penis diameter is increased by thickening both the cuticle and the muscles. When barnacles reside in areas of lighter wave action, those same species of barnacles were observed to have longer, thinner penises. Studies have indicated that the length of the penis varied as much as 25% longer than those living within heavily exposed environments. However, even though the diameter or length was found to be thickened, the overall mass of the penis did not increase. When distance between mates became larger, barnacles evolved longer penises in order to reach further distances by adding more annulations. Many experiments have shown that with these morphological adjustments, barnacles have been able to increase their mating success. And phenotypic plasticity is not only used to increase sexual success, but can also increase success in other aspects of the individual's life. Barnacles' cirri are also receptive to phenotypic plasticity. Individuals have adapted a long, thin feeding cirri in areas with little to no wave action, and short, thick cirri in areas where they are often exposed to wave action. These morphological adaptations help the individual to be better suited to withstand harsher environmental conditions, as well as living within crowded or secluded areas. So, Chelonibia testudinaria is the target species of barnacle being researched within my thesis, and Chelonibia is an epizootic barnacle, meaning that it is a species of barnacle that lives on the surface of another living organism with relatively no harm, like this turtle here. And this relationship between the host and the surface dweller is considered commensal. And epizootic species have been found to date back to 5 to 15 million years, and examples of that include Porifera, Cnidaria, Mollusca, and Arthropoda. Barnacles are so commonly found upon sea turtles that more than 94% of nesting loggerhead sea turtles host at least one species of barnacle at any one time. And turtles have even been observed to have Chelonibia testudinaria year-round. The benefit barnacles receive from being on the backs of mobile hosts include the greater potential for nutrients to be gathered due to the host creating water flow by moving. And Chelonibia is one of the most common genus of acorn barnacle residing on mobile hosts. And within this genus of Chelonibia, there were formerly 3 recognized species, differentiated by the host that they reside on. However, the species of barnacle were genetically indistinct from one another, so all 3 are recognized now as Chelonibia testudinaria. Chelonibia testudinaria is incredibly host-specific and rarely resides on anything other than sea turtles, which is why they are commonly referred to as turtle barnacles. Chelonibia testudinaria can reach a rostral carinal length of 120 millimeters. The rostral carinal length is the distance from the most anterior plate, the rostrum, to the most posterior, the carina, also known as the total length of the barnacle. And Chelonibia grows linearly, growing 0.07 millimeters per day in rostral carinal length. Chelonibia grows until it is removed from its host due to the turtle's carapace molting, host removal by rubbing against a hard substrate, or scientific collection. It is typical for epizoic barnacles to be removed due to molting. Sea turtles' individual scutes molt in bits rather than full sheets, meaning that molting can be a long process, allowing for Chelonibia to grow very large. Because of this, it makes lifespan very difficult to determine for Chelonibia testudinaria. However, it is safe to say that the larger the barnacle, the older the individual is compared to surrounding barnacles. Little information regarding Chelonibia's life cycle has been discovered. This includes breeding cycle and frequency of reproduction, and the lack of information is likely due to the fact that it is incredibly difficult to monitor these barnacles that reside on free-moving mobile hosts. And turtles being an endangered species require strict permitting to work with, and this alone is a difficult requirement to gain access to. It is also incredibly difficult to monitor the same individual within a natural setting, to regularly check up on these hosts that migrate freely and at great distances. Because of this lack of information regarding the life cycle of Chelonibia, I'm going to briefly talk about the life cycle of barnacles in general. So, typically, larval development within barnacles includes 7 instars, or molting periods. The first 6 instars are called napulae, which are unsegmented larvae, and the first few may reside within the mantle of the adult barnacle before being released into the water column with the rest of the plankton. The 7th is referred to as a cyprid, or the last larval stage before adulthood, whose main priority is to settle in an appropriate area rather than feeding. For these turtle barnacles, it is a difficult task for the newly settled individuals to succeed, as they need time to settle onto their host and find proper nutrients before the host migrates to areas that may be less productive. For instance, juvenile loggerhead turtles spend upwards of 6 years in productive oceanic nursery grounds, whereas older individuals who have left the nursery phase travel on large seasonal migrations, which include areas of patchy productivity. The most abundant place that turtle barnacles attach to is the carapace, followed by the plastron, and then less commonly found on soft body parts like the turtle's neck, flipper, or their tails. Turtles provide a patchy environment for barnacles to settle, and this tends to mean that Chelonibia typically live within clusters called hummocks. Typically, barnacles reside within these clusters at more optimal locations. However, optimal locations on the turtle's carapace may not be the easiest place to settle as a cyprid. It is likely that cyprids find a suitable location to settle, but end up moving as an adult barnacle to find an ideal position for feeding and reproduction, caused by water flow across the shape of the turtle's carapace. Chelonibia has been observed to migrate across their host carapace to find a more preferred location, and this is incredibly unique when talking about an otherwise sessile organism. Optimal spatial distribution is determined based on water flow as well as inter- and intra-species competition. And water flow is a key factor to consider as it affects all stages of the organism's life, from dispersal to recruitment as well as overall survival. This optimal distribution appears to be on the posterior portion of the carapace, determined by other studies focusing on Chelonibia's spatial distribution. And this is due to the posterior scutes having a lower water flow as compared to that of anterior scutes. Some other studies have shown that barnacles were more abundant in central scutes, but when it comes to looking at overall size distribution, specifically on loggerhead turtles, they tend to have the larger barnacles aggregating towards the anterior half of the carapace. And green sea turtles that were observed tended to have a majority of their barnacle coverage on the marginal scutes. Central and marginal scutes could be favored due to the ability to keep sediments and food particles more effectively due to the flow patterns eddying around unique carapace features found on those central and marginal scutes. Knowing how epizoic organisms spatially distribute amongst their hosts provide important indicators into movement patterns, migration routes, geographical distribution, as well as health of the host they are attached to. This is not only pertinent to barnacles on the backs of sea turtles, but a slew of epizoic organisms and the information they can provide us about their hosts they reside upon. Although turtle barnacles are widely known, research being conducted on them is scarce due to the difficulties in accessing them. And this study presents information vital to the understanding of how Chelonibia adapts to a multitude of variables within a natural setting. Specifically, my research aimed to answer several questions. The first being, does the barnacle's anatomy of penis and cirri vary with body size? From this question, I would expect that barnacles with a larger body would have larger appendages compared to smaller barnacles. Question number 2. How does barnacle position on the turtle affect the morphology of the penis and feeding cirri? And from this, I would expect that barnacles residing within certain portions would have altered morphological features compared to individuals within different positions. For instance, anterior barnacles are susceptible to a higher water flow rate compared to those on posterior scutes, whose water flow rate is drastically lessened. Because of this impact, I expect that in order for barnacles to withstand these different conditions, their morphology would need to be altered. Thicker appendages in order to withstand harsher flow rates. Question 3. How do barnacles more distant from each other differ in the morphology of the penis and feeding cirri compared to barnacles growing within a cluster? And from this, I would expect that social condition would be a contributing factor to why barnacles have adjusted their morphology. The further away the potential mate is, the longer the barnacle's penis must be to extend in order to fertilize the egg masses. Similarly, those individuals within tight clusters may not need such drastically long penises. Cirrus morphology would also be affected due to social condition, as crowding tends to make filter feeding more difficult. Question 4. How does social condition affect fertility probability? And from this, I would expect that social condition does impact fertility probability, and it is more likely for individuals close to a potential mate to be fertilized or fertilize a closer potential mate themselves, rather than those at a further distance from one another. And lastly, question 5. What is the difference in spatial distribution of barnacles on the carapace of Caretta caretta, or loggerhead sea turtles, compared to Chelonia mitis, or green sea turtles? And I expect that barnacles observed within this research would be found in greater numbers on posterior scutes, as optimal positions for barnacles have been suggested to be on the posterior portion of the turtles. Overall, the significance of this research is to better bridge the knowledge gap there currently is within such a commonly found and important species of epizoic barnacle. And I answered these questions by describing Chelonibia testudinaria distribution and abundance from turtles found on beaches in Broward County, Florida, collected in the years of 2019 and 2020. So since Chelonibia testudinaria is a species of epizoic acorn barnacle that utilizes sea turtles as their permanent residents, nesting loggerhead and green sea turtles observed off of Fort Lauderdale Beach were used within this research. The removal of epizoic barnacles was already a sampling protocol integrated into the turtle tagging program run through Nova Southeastern University's Conservation, Movement, and Ecosystem Dynamics Laboratory, so the only adjustment to this protocol was to preserve the barnacles removed from the carapace of the nesting turtles, rather than immediate disposal. And nightly patrols span from 2100 hours to 0500 hours on most nights, weather permitting, where loggerhead sea turtle nesting period is in Florida ranges from April to September with peaks in June and July. And patrols were designed to look for nesting and false crawling female loggerhead and green sea turtles on the beach. Once a turtle was spotted, the turtle was observed. This was to determine if she was on her way to nest or returning to the ocean. And once a turtle was determined to be on her way back to the ocean, a corral was placed around the turtle to allow for sampling to occur. The turtle was then named and numbered for cataloging. The carapace was then cleaned with seawater to allow for more clear photos from the top down to be taken of the barnacle's positioning. Close-up images of the barnacles clusters were also taken to determine individuality of the barnacles before being removed and potentially separated from those clusters. And barnacles were then carefully removed using a chisel or paint chipper, dependent on the size, location, and structural soundness of the turtle's carapace. Removed barnacles were then placed inside of a containment bottle, rinsed with seawater, then preserved with ethanol to be transported back to the lab. Each individual barnacle collected was compared to the top-down photos taken prior to removal, and these photographic images were used to determine individuality and position each barnacle originated from. Barnacles were determined based on unique exterior carapace features, given a number, and separated into individual containment bags filled with ethanol. Each turtle's carapace was separated into 4 different positions: anterior, posterior, central, and central coastal. And these positions were used to determine spatial distribution barnacles had on the carapace of sea turtles surveyed. Not all of the barnacles were able to be removed from the host for a variety of reasons, including potential harm to the turtle's carapace, as well as structural damage to the barnacles upon removal. For this, all barnacles that were photographed rather than collected were used to determine spatial distribution on the carapace of turtles surveyed. After individuality and position was determined for sampled barnacles, each barnacle was weighed to determine total weight. The soft tissue was removed from the barnacle's carapace, where soft tissue weight was determined, and measurements of shell height, rostral carinal length, base length, and aperture length were recorded using a digital caliper. These measurements were used to determine volume of the barnacle shell, and the shell's volume was determined based on the formula for a truncated cone, which is shell volume is equal to 1/3 times pi times h, parentheses, small r squared plus small r, times big R plus big R squared, where h is the height, big R is the radius of the basal opening, and small r is the radius of the aperture. Once the barnacles were properly identified and measured, they were dissected. Many intertidal barnacles have a hard base plate to protect the internal soft body tissue from the substrate they attach to. However, Chelonibia testudinaria does not possess this base plate. Instead, Chelonibia has a slightly opaque film on the dorsal sector of the organism, and to get to the internal soft body, the film was removed using forceps, where their internal structure was carefully removed from the protective housing, weighed to get an internal mass, and observed to be carrying eggs or not, then placed under a dissecting microscope to properly examine the individual's sexual and feeding structures. Series 1, Series 6, and the penis were removed from the body to be placed onto slides in that respective order from left to right across one slide per individual, and these slides were covered in preparation for microscopic photography. A Hyrox digital video microscope was used to photograph the small internal structures removed from each individual barnacle, and again, those structures include Series 1, Series 6, right, and the penis. So Series 1 and Series 6 were measured for total length from base to tip, basal thickness, or the diameter of the base, as well as the number of segmentations along the Series branch. And each penis was measured for total length, basal thickness, as well as number of annulations. Once each structure was photographed, those photographs were uploaded to a computer to analyze using the imaging software of ImageJ. And ImageJ uses known distances accounted for by the calibrated scale on the digital video microscope and converts the unknown pixel length into an accurate measurable distance. This was incredibly useful when determining total lengths and basal thickness. However, counting segmentations and annulations would require a more laborious strategy. I was fortunate enough to come across a very useful tool ImageJ offers within its software, and that is the plugin feature. It's a way to add packages to the software that may not already be accessible in the initial download. Doing some searching online, I had found a plugin program that was used to count tree rings autonomously, and I figured it doesn't hurt to try. Tree rings and annulations seem to be similar enough, so that's what I ended up doing. And I was able to modify this tree ring specific plugin to aid in counting annulations of barnacle penises. For the most part, this program was accurate. However, with the sheer number of annulations and how close together they were, the plugin missed a few. So after the program concluded, a manual eye correction was needed to add in the few annulations the program may have missed. Within my research, questions being asked were statistically analyzed by means of determining how correlated each variable was to one another, as well as a factorial analysis of covariance, or factorial ANCOVA, and by means of creating a logistic regression. Spatial distribution was analyzed using a simple chi-squared test, and all statistical analysis was run using the program R. A factorial ANCOVA was used to test interactions between position and crowding and the measured data collected, and a covariate was used within this analysis. A covariate is a variable that may potentially impact the results of the test, and within my research, the covariate used was body size, specifically the internal weight of the barnacle. An internal weight was used as a representation of body size to remain constant with similar works. And each measured dependent variable was tested against two categorical variables using body size as the covariate. The dependent variables measured using this test include penis diameter, measured penis length determined by counted annulations, serous diameter, measured serous length, as well as serous length determined by counted segmentations for both Series 1 and Series 6. The two categorical predictor variables being tested included position the barnacle resided on the turtle's carapace, as well as whether the barnacles grew within a cluster of other barnacles, or if the barnacle was considered solitary. The reason why both variables were used within this analysis to create a factorial ANCOVA, rather than doing multiple simple ANCOVAs, was to ensure that the significant interactions between the categorical variables and the dependent variables were unique and not improperly determined significant when other variables were actually the determining factor. The next analysis I ran was a logistic regression, and the purpose of a logistic regression is to help predict the likelihood of a variable changing based on one or more independent variables. And within my research, a logistic regression was run to predict the likelihood of a barnacle containing eggs within a clustered area. The dependent variable was whether the individual barnacle was observed to have eggs within its inner mass, represented as a 1 value, or if the individual barnacle was not observed to have eggs within its inner mass, represented as the 0 value. The independent variables used to predict the likelihood of barnacles containing eggs was crowding as well as internal volume of the barnacle. Internal volume was used rather than either measurement of mass because body weight would typically be heavier for individuals observed to have eggs compared to those of smaller size not observed to have eggs. Lastly, a chi-squared test was conducted to determine whether barnacle distribution was distributed based on the expected outcome by means of surface area coverage, or to determine if there was a relationship between position on the turtle's carapace and barnacle distribution. As I had mentioned within the methods portion of my defense presentation, I had run some correlation tests to determine how correlated each measured variable was with one another. And this was helpful in determining which measurement could properly be used as an accurate representation of a barnacle's body size. Within this test, the closer the value is to 1, the higher those variables are correlated. The closer the values are to 0, the less correlated those values are. And for values close to 0.5, those variables are considered slightly correlated to one another. Looking at the body size variables, it is clear to see that all variables are highly correlated to one another, meaning any variable can be used as an accurate representation of body size. And this is beneficial in the sense that I used internal weight as my covariate for my factorial ANCOVA, and similarly used internal volume when conducting logistic regression. I was able to use each variable as they fit best for each test without sacrificing an accurate representation of body size. When looking at the measured variables for the barnacle's penis morphology, it makes sense for penis length and penis annulation to be highly correlated, as both of those variables represent length of the penis. However, it is still important to measure both data since barnacles have the ability to extend and retract their penis. Both variables, highly correlated in all, provide a better understanding of how penis morphology and the variables that affect it. Penis length and penis diameter are also slightly correlated, which makes sense as in some cases individuals within different environmental conditions were found to have longer, thinner penises, meaning that those variables would be somewhat correlated. Penis annulations and penis diameter, however, were not correlated within this study. Compared to the penis morphology, series morphology followed a somewhat similar correlation trend. Length and segmentations were highly correlated. Segmentation and diameter were slightly correlated, but a major difference was that length and diameter were highly correlated for both Series 1 and Series 6. All of the measurements appear to be correlated to one another, and this is likely due to the fact that 1. The cirri do not have annulations but segmentations, meaning that they do not extend and retract to a longer length. Or 2. The elasticity of the structure is lacking, meaning that the structure's overall measurements are likely predetermined rather than having more phenotypic elasticity. So, this factorial ANCOVA tested all measured variables for penis morphology as well as all measured variables for cirri morphology, compared to clustering and position on the turtle's carapace using internal weight as a covariate. And starting with penis diameter, the barnacle's penis diameter was determined to be strongly positively associated by both body size of the barnacle, as well as if the individual was found within a cluster or considered solitary. Position on the turtle's carapace, however, was not considered a significant predictor of penis diameter. And the figure seen on this slide shows the significance clustering has on penis diameter, where crowded individuals are represented by orange circles and solitary barnacles are represented by blue triangles. And position was excluded from this graph as there was no significant interaction between these variables and position. Penis length was also positively associated with the barnacle's body size. Position on the turtle's carapace also had a positive association with penis length, along with the interaction between position and clustering variables and the interaction between position and body size. Within the interaction of position and clustering, there was a significant difference between individuals within central costal scutes and anterior scutes. And within the central costal and anterior scute grouping, there was a significant difference between clustered and solitary barnacles. Individuals that were within a cluster on central costal scutes had a significant different penis length compared to individuals within a cluster on anterior scutes. And individuals within a cluster on central costal scutes also had significant different penis lengths compared to barnacles living solitary on anterior scutes. The rest of the groupings were determined to not be significantly different from one another. And clustering as a whole was not significantly associated with penis length, as well as the interaction between clustering and body size. The significant interactions on the figure include the clustered barnacles on central costal scutes, which are represented by green squares, clustered barnacles on anterior scutes, which are represented by red circles, and solitary barnacles on anterior scutes represented by blue open squares. Penis annulation was positively associated with body size and position. The barnacle resided on the turtle's carapace. However, for this variable, position had a more general effect on penis annulations, and there was no strong difference between any of the positions against one another, and that is clear by the figure created above. Penis annulation was also not significantly different between clustered and solitary individuals, where clustering was not included within this figure, and there was no significance. So I separated these results into Series 1 morphology and Series 6 morphology, rather than keeping it more generalized as cirrus morphology. And the reason for this is due to each set of cirri having different roles within the barnacle. Barnacles may exhibit phenotypic plasticity for one set of cirri, but not the other. And this is because the barnacle's cirrus function may need to be adjusted due to environmental conditions, whereas those conditions may not affect the other roles the previous cirri perform. Series 1 is mainly used for grooming and holding onto food particles for consumption, whereas Series 6 is used for filter feeding. Series 1 diameter was positively associated with body size as well as crowding. Position did not have a significant association with Series 1 diameter, and the figure shows the difference crowding had on Series 1 diameter, where crowded individuals were represented by red circles and solitary individuals were represented by blue triangles. Series 1 length was significantly associated with body size, where both position and crowding did not have a significant association with Series 1 length, and the figure above shows a lack of significant difference between crowded individuals and the barnacles residing within different positions on the turtle's carapace. Series 1 segmentations were positively associated with body size, as well as position on the turtle's carapace. The difference between individuals residing within the interior portion of the turtle's carapace and central scutes, as well as the individuals within central costal and posterior scutes, were found to have a significant difference between Series 1 segmentations. The interior position is represented by red circles, the central position is represented by green triangles, and the central costal position is represented by blue squares. And the posterior position is represented by purple crosses. Crowding was not significantly associated with Cirrus 1 segmentations. Cirrus 6 diameter was significantly associated with body size. Position and crowding did not have a significant association with Cirrus 6 diameter. However, there was a significance between the interaction body size has on both position and crowding. And the figure just better shows the lack of significance position and crowding has on Cirrus 6 diameter. Cirrus 6 length was positively associated with body size as well as position on the turtle's carapace. Specifically, Cirrus 6 length was different for individuals within the posterior portion compared to those within central coastal scutes. And the posterior position is represented by purple crosses, and central coastal scutes are represented by blue squares, where crowding did not significantly affect Cirrus 6 length. Cirrus 6 segmentations were positively associated with body size as well as position on the turtle's carapace, where Cirrus 6 segmentations were significantly different for individuals within the posterior portion compared to those within the central coastal scutes. The posterior position is represented by a purple cross. The central coastal scutes are represented by blue squares. Crowding did not have a significant effect on Cirrus 6 segmentations. So I know that was a lot, so I just wanted to go back through and go over the overall implications of what all of those graphs mean. So body size was positively associated with penis morphology, such that the larger barnacles had larger penises. And this is to be expected. Typically, larger individuals possess larger penises in all facets, diameter, length, and total annulations, than those of smaller body size. And that's because a bigger body allows for larger appendages to be housed. And this goes for Cirri morphology as well. A larger body supports larger appendages, sexual and feeding. So, Chelonibia's penis morphology was also associated with position on the turtle's carapace. The only variables that were associated, however, were for penis length. Both measured and annulations were associated with position on the host. And the difference between positions occurred within barnacles residing within anterior portion of the carapace and barnacles residing on central coastal scutes. Barnacles residing on central coastal scutes had a longer penis length, and on average they had a longer penis of 27.27% than those within the anterior portion of the turtle's carapace. A few possible explanations for why barnacles within the anterior portion of the host's carapace would have shorter penises include age as well as sexual status. The individuals within the anterior portion could be smaller and therefore younger than those residing on the central coastal scutes. The interior portion may also be a better recruitment site for newer barnacles to settle, meaning the younger barnacles would have a smaller penis compared to older, more established individuals. It is suggested that barnacle larvae could favor certain positions due to environmental conditions such as water flow. This is supported by barnacles being 14.74% larger on average within central coastal scutes compared to those on interior scutes. It is also interesting to note that penis diameter did not change based on position. It is common to see intertidal barnacles have penises with larger diameters in areas of higher wave action, compared to barnacles less exposed. Making assumptions about water flow patterns on the turtle's carapace, I had hypothesized that some positions would require a larger penis diameter to combat the differences in water flow. And seeing how position on the turtle's carapace was not a significant associated with penis diameter, we can hypothesize that the difference between flow patterns for each position on the turtle's carapace is not harsh enough to require phenotypic plasticity. Another theory on why barnacles do not have a difference in penis diameter amongst position is that perhaps it is more energy efficient to move to a preferred location rather than adjusting its penis morphology. The barnacle may not plan on being in the recruitment site for too long, and adjusting its morphology may be an unnecessary energy expenditure. Clustering was not significantly associated with penis morphology, and this was not expected again. You would tend to expect that individuals at a greater distance to their closest mate would require a longer penis to fertilize their eggs. And similarly, it is expected that individuals of close proximity would be able to support a much shorter length of penis to properly fertilize potential mates without wasting energy. There was only one significant social condition had on penis morphology, and that was within its interaction with position when analyzing penis length. And it is safe to say that position is more likely more significantly associated with penis morphology rather than social condition. Overall, Cirrus morphology was associated with both position and social condition, where position, however, only impacted segmentations for Series 1, and the difference was between anterior barnacles compared to the rest of the positions. Anterior barnacles had less segmentations compared to the other 3 positions, and this is interesting that the segmentations were smaller, but the overall length didn't change. Similarly to the penis morphology, it is likely that anterior barnacles are younger, meaning that they have not developed enough to have their segmentations on par with the rest of the community. Because Series 1 is the shortest, it could make sense that the overall length is not significantly affected, as the level of difference is likely on a smaller scale. Social condition affected Series 1 diameter. On average, crowded individuals had a thicker Series 1 diameter, and this could be due to the individuals within close proximity to one another attempting to obtain any advantage they can when it comes to feeding against close competitors. Where crowding tends to make filter feeding more difficult, and solitary individuals may not require a thicker cirri as feeding may be more readily available. Another possible reason for the significance could be within hummocks. The water flow patterns are higher, requiring sturdier appendages. Position was significantly associated with both length variables for Series 6, and the significance between positions was found between posterior barnacles compared to central coastal barnacles, and on average, length was longer for barnacles residing on the posterior portion compared to those on central coastal scutes. And this difference is likely due to flow patterns across the turtle's carapace. Barnacles residing on the posterior portion of the turtle's carapace experience less water flow, allowing for a longer cirri to be supported. Longer cirri would also suggest that the individual must extend further to obtain their nutrients, and perhaps individuals residing on the posterior portion of the turtle's carapace do not receive the proper amounts of nutrients that other positions would receive. This would suggest that posterior barnacles exhibit some form of phenotypic plasticity when it comes to feeding. Recapping the setup for logistic regression, fertility was categorized by a 1 for having eggs, or a 0 for not having eggs. Other variables being used within this regression included internal volume as well as social condition, or whether the barnacle was within a cluster or considered solitary. And internal volume did not appear to have a significant effect on predicting fertility. However, social condition was a significant factor in predicting fertility. Keeping all variables constant, when crowding increases by 1 unit, the barnacle is 0.679 times more likely to have eggs as opposed to not. And in other words, odds of being fertilized within a crowded community is 68% times higher than not being fertilized. This statistic makes sense because it is a lot less likely to be fertilized within a solitary community. As for internal volume, it would make sense for volume to help predict barnacles being fertilized, as the larger the volume, the more space available for egg production. However, this is just an assumption when in fact there are other variables to consider when determining fertility probability, like proximity to potential mates and penis size. As for the figures displayed, the first is an actual logistic regression, and this figure shows the lack of relationship that volume has on predicting fertility. As for the other figure, it shows the likelihood barnacles have been fertilized based on social condition, showing that crowded individuals are more likely to be fertilized than barnacles that are considered solitary. So, all barnacles observed within the photographs taken on collection night were used within determining the spatial distribution for this research, rather than just using the barnacles collected and dissected. And this is to create a more accurate representation of overall spatial distribution on the carapace of loggerhead and green sea turtles. Each turtle's photo was separated into 4 generalized scoop positions, and those positions again were anterior, posterior, central, and central coastal. Loggerhead sea turtle barnacle distribution was highest on posterior scutes, followed by central, central coastal, and lastly being the anterior portion of the carapace. And as for green sea turtles, barnacle distribution was highest on central coastal scutes, followed by central, anterior, and lastly the posterior portion. It makes sense for the posterior portion of the turtle's carapace to be a more populated area, as it has been suggested that the optimal position for barnacles would be on the posterior portion of the carapace. And this is the location barnacles have been observed to migrate to. However, spatial distribution appears to vary based on species of turtle. And a few explanations of this include the sample size could be way off. For instance, 30 loggerhead sea turtles were sampled from, and only 3 green sea turtles. Typically, the green sea turtles that were observed to have been nesting did not have many or any barnacles on their carapace. Another possible explanation could be that the different species of turtles have different optimal positions. Flow patterns eddying around unique carapace features for each different species of sea turtle. And it is also possible that the green sea turtles who were observed to have fewer barnacles could have a better way of removing epizoic barnacles. And lastly, these barnacles could be younger and have not had enough time to move to a more desirable location. Included are the chi-squared and p-values, showing that these barnacles are distributed differently from what was expected based on percentage of area coverage. And this analysis backs up the fact that there is a relationship between position on the turtle's carapace and barnacle distribution. So, in conclusion, this research yielded some very interesting results. Some similarities between Chelonibia testudinaria and other acorn barnacles were expected. However, there are some key differences between the species. These barnacles appear to not be significantly affected by many variables we would expect when living on a mobile host. And intertidal acorn barnacles exhibit phenotypic plasticity based on many environmental factors. Wave action and crowding significantly affect the penis and cirrus morphology amongst intertidal species. However, similar variables did not have such a drastic change on Chelonibia. Intertidal barnacles had thicker structures in areas of greater wave action, but when it came to Chelonibia, these barnacles were not as affected by more dynamic environmental conditions. Barnacles residing within the interior portion of the turtle's carapace experienced the highest level of flow, similar to barnacles residing on or around the tide line. These individuals collected from the interior portion of the turtle's carapace did not have thicker sexual or feeding structures compared to areas of lesser drag. Knowing that Chelonibia is not completely sessile and can move to preferred areas with less water flow, it is safe to conclude that modifying the structural soundness of the barnacle's appendages is not worth the energy. The biggest impact is indeed the host they live on, where host movement creates water flow, allowing for the potential to obtain a greater amount of nutrients. No matter where these barnacles reside on their host, theoretically, they should have access to plenty of food particles without needing to adjust their structural morphology. However, whenever the individual reaches its preferred location, Chelonibia testudinaria adjusts its morphology. Barnacles residing on the posterior portion of the turtle's carapace, an area of lower water flow, exhibited phenotypic plasticity when it came to their cirri. Barnacles had significantly longer cirrus 6, the active feeding cirri. Similar to intertidal species, the expectation for longer appendages in areas of less flow is indeed met. In areas of clustering, more difficult areas to filter feed, the only significant adjustment barnacles made was a stockier cirrus 1. A thicker first cirrus allows for barnacles to better hold on to food particles in areas of eddying and greater feeding competition. Combining all of the information obtained from this thesis, we can conclude that the interior portion of the turtle's carapace is more readily available for a recruitment site for larval Chelonibia. Barnacles found within the interior portion of the turtle's carapace tend to be on average smaller than other positions, and were determined to have a penis 27% times smaller than those on marginal scutes. It's also interesting to note that dwarf barnacles were not overly abundant within this research. Looking at how dwarf barnacles impact potential mates within a community of Chelonibia testudinaria was one of the main goals when originally designing this research. I feel like dwarf barnacles are an important member of Chelonibia communities, and thought it was interesting to have such a sparse population being collected. This was certainly not the expectation when conducting this research. Ultimately, this project aided in closing a significant gap in scientific knowledge pertaining to these tough-to-observe barnacles. We now have a better understanding of how barnacles congregate on the back of mobile hosts, and how barnacles' locations affect barnacle morphology. Barnacles that reside on the backs of moving hosts face environmental factors unique to these epizoic organisms, and water flow patterns change based on the water column the host resides in, as well as these barnacles are at the mercy of their hosts when it comes to food particle collection. These epizoic barnacles combat difficult environmental factors by means of phenotypic plasticity, as well as moving to a more beneficial location on the host. This research offers an important understanding of how organisms may adapt to distinct environmental factors differently from what may have been expected. Many organisms adapt phenotypic plasticity when impacted by different environmental factors, and this species of barnacle provides an unprecedented understanding of how sedentary species adapted to move locations rather than adjusting their structural morphology. Perhaps other sessile invertebrates have adapted different means of conserving energy when it pertains to adjusting their environmental constraints. Plans for future research include attempting to determine which individual barnacle may be the potential parent for fertilized eggs, and how position potentially affects successful mating. I would like to express my appreciation firstly to my amazing major advisor, Dr. J. Matthew Hoke, who reached out to me during my struggles throughout this graduate program. He ultimately helped me through my journey of mastering the marine sciences. He continuously aided in scientific discussions inside of the lab as well as out in the field. There was not a moment in time where I felt like I was alone during this process, and for that I thank you. Without you, none of these findings would have become possible. I would also like to thank my wonderful committee comprised of Dr. Joshua Feingold and Dr. Omar Eldakar for their guidance, especially when it came to the writing process of this thesis paper. Their expertise in invertebrate biology as well as the evolutionary sciences played a key feature in the completion of my journey. I'd like to thank the Sea Turtle Ecology Lab at Nova Southeastern University, Dr. Derek Burkholder, as well as Glenn Goodwin for his ability to help during the collection process, as well as taking me under his wing during sea turtle tagging season. I truly would not have a project without being able to collaborate alongside with his sea turtle nesting study. I'd like to also express my deepest gratitude to my family and friends who have been there for me after long days and even longer nights. Rhonda and Guy, thank you for being the best parents a son could ask for. Thank you for being one call away as I found something weird or interesting I wanted to talk about. I always knew I could count on the two of you to listen to me as I rambled on about my research. Ultimately, thank you to the people who have supported me throughout my graduate school experience. I truly would not be able to do it without each and every one of you. Any questions?